Hello, everyone. I'm Mike Levy. I organized the Institute for Neural Computation Chalk Talk series. I invited for our first Chalk Talk this semester, Tim O'Leary, who I met at SFM quite a few years ago. He started a PhD in math, wound up postdocing with Eve Martyr for a while, and now it's been at the University of Cambridge in the engineering department, I believe, for a few years. And he's going to talk to us today about representational drift. Thank you for speaking with us today.
Thank you, Mike. Okay, I'm going to jump right in and define this phenomenon, representational drift, starting with experimental observations. So probably many of you are aware that across brain regions, so-called task representations seem to continually evolve without obvious changes in task-related behavior. A famous example of this was published by Yaniv Ziv in Mark Schnitzer's lab more than a decade ago. And in this study, mice were trained, or motivated rather, to run back and forth along a linear track. And they did this while they had optic fiber microscopes implanted in their brains so that Ziv could image CA1 place cell activity. And he discovered there's very nice place fields. So if we look at cells as a function, so this is normalized calcium activity as a function of track location across 100 or so cells, then we see these nice localized bumps of activity in various locations of the maze. And these cells have been sorted in such a fashion that the peaks are arranged from the beginning of the maze all the way to the end. So we have this nice tiling of the maze. That, by the way, is loosely what we mean by this population representing behavior. What Ziv then observed was that this same population on later days in the same task with no obvious change in the animal's behavior had markedly different activation properties. But it wasn't the case that the cells simply became unresponsive to locations in the maze. On any given day, it was possible to identify neurons in the same field of view and order them such that you could recover this tiling of the task. Similar work some years later in Chris Harvey's lab by Laura Driscoll studied posterior parietal cortex, this time in a head-fixed virtual reality task where the animal has to do something a bit more interesting. It's shown a visual cue at the start of the T-maze. It has to remember that cue during a delay period, and then it has to turn left or right according to the identity of the cue. And mice learn this very well. They become experts at this task and they can repeat it hundreds of times in a session. So using this task and 2-photon calcium imaging this time, Laura studied the activation patterns of posterior parietal neurons and found largely the same thing. So there are indeed activity patterns that tile the task. These, if the ordering of cells is maintained across days, then after 10, 20 days, the activation becomes less obvious. Yet on any given day, by reordering cells, it's possible to recover this nice tiling of the task. And I'm showing here examples that we replotted of some of these units, just so that you can see what goes on in a random selection of 3 different cells. So here we see a unit that's active, that's active here at the start on day 1, at the beginning and the end of the maze location, and it gradually acquires this prominent field toward the end, toward the midpoint of the maze. This unit, by contrast, has relatively stable tuning. So it's not the case that all of the cells reorganize, but a large number of them do. So interestingly, evidence of this representational drift preceded these papers by a decade or more. But convincing evidence of it had to wait until one could identify cells unambiguously and track them for many days, and that's experimentally challenging. Prior to this work, at least a decade before this work, the state-of-the-art involves tetrode recordings and source localization by spike sorting. And Cliff Kentros published a paper back in 2004 showing that again in CA1 in place cells, this time in a circular environment, across recording sessions, which sometimes correspond to across days, the same cell has a very prominent firing field, which appears to reconfigure. So each row here is a different cell, each column is a different day, and we can see clear place fields, although the location of the place field is not consistent over time. Now, interestingly, this was interpreted in a different way in this particular study. And the interpretation is nicely captured in 2 sentences by the senior author of this work, Eric Kandel, who in a fairly influential review mentions that, I'm sorry, I think I have the date wrong here, that when a mouse does not attend to the space it walks through, the map forms, but is unstable after 3 to 6 hours. When the mouse is forced to attend to the space, however, the map is invariably stable for days. So the examples I'm showing here, the interpretation at the time of publishing was that the mice were not forced to attend to the environment sufficiently. Their mind was kind of wandering, and that somehow explained the movement of these place fields. And then in a modification of the task, where mice were required to attend to various locations, they found more stable responses over a few days. So all of this work, especially the later work where there could be no real doubt that animals were attending to salient parts of the task, yet there was still drift, is completely consistent with the idea that neural circuits have a very large configuration space. And in particular, the activity patterns have a large coding space. And there are many valid patterns, if you like, that could correspond to various task variables and so on. So we can think of this configuration space as being a big manifold. And as long as configuration is in or close to this space, the circuit functions essentially, or a memory is faithfully recalled. And we can think in this view of learning or some form of reinforcement or feedback as driving the circuit configuration into this space, which is learned. And we're going to draw on this geometric picture quite a lot at an abstract level. And immediately, it's clear that if we have these degenerate configurations and something which disturbs a configuration, but some kind of continual feedback that maintains the circuit within this allowable space, then this is a recipe for generating drift. So we might say that degenerate circuit configurations, which we know that these are a feature of neural circuits, plus some form of continual feedback, in some sense predicts representational drift, provided there is something to cause the circuit to reconfigure slightly over time. So in this talk, what I'm going to lead you through is ideas about how drift can reveal properties of circuit plasticity and neural representations. So first, we're going to talk about how to relate drift to systematic plasticity and continual learning. Second, how drift can be used to quantify degrees of freedom in the task representation. And third and finally, we'll ask whether features of drift, so empirical features of how cells reconfigure their firing properties, whether these are indicative of some kind of underlying plasticity mechanism. And by the way, if at any point any of this is unclear, do interrupt me for a clarification. Okay, so let's begin with this first question of relating drift to some form of systematic plasticity. And I'm going to change scales a little bit and start with another observation made around the same time. And that is that steady state synaptic change in the cortex of intact behaving animals is surprisingly high, even when the animals aren't overtly learning. So this is a paper from Simon Rumpel's lab with Jonathan Lewinstein, who tracked the motion, appearance, and disappearance of protrusions along the dendrites of principal cells in the auditory cortex over many days. Again, it's a challenging experiment, and it kind of had to wait for the technology to be there to do this. And this was during a learned auditory task. And what they found is that over a period of several weeks, the spine population was essentially replaced. 50% of the population was replaced by newly born spines, and the existing spines died off. So there was this continual turnover, if you like, of new spines and old dendritic spines disappearing. And they took this as evidence that the connectivity in the cortex was also changing significantly. And there's been other studies that have illustrated this and checked essentially whether these putative synaptic contacts are in fact functional contacts. And we've got reason to believe that they are, and that this type of turnover is functional. And other kinds of experiments have attempted to separate out systematic changes from random fluctuations. And I'll try to explain what that means. Here is a paper which I'm just showing the summary of by Dworkin and Noam Ziv, published in 2016. And in this paper, this was actually conducted in neural culture, in slice cultures. And what the authors did was essentially build a large regression model where they tried to account for changes in spines to various other observed processes. So remodeling of dendrites or activity history in the adjacent dendrite or the cell body. And in this way, they were able to account for some of the turnover of spines, quite a bit of it, but there was a large percentage that seemed essentially synapse autonomous. So for no apparent detectable reason, the level of the cell or the cellular physiology, these spines seem to come and go quite a bit. And they aren't the only authors to observe this kind of proportion. Other studies have attempted to either block plasticity or in some way account for changes during or after behavioral enrichment to try and manipulate spine dynamics. And a really common finding is that the vast majority of the flux in spines seems to be something that can't be controlled by either pharmacological blockade or behavioral manipulation or other kinds of physiological correlates. So if we take this at face value, this tells us that synaptic fluctuations contribute more than 50% of total synaptic change, and something less than 50% seem to be these systematic changes. So we can ask lots of questions about this. Are there imaging problems? Is it the model system, etc.? We will take the results at face value. It's not clear why the systematic changes are so weak. Perhaps this is just the best that biology can do, but that seems tremendously wasteful energetically. And what we're going to claim is that the optimal ratio for maintaining a learned behavior is in this range, in fact. And that this proportion scales not only that this makes sense, but this proportion scales with learning rule precision. Excuse me, is someone?
Yes, I have one question. What do you refer when you say the systematic changes?
I'm sorry, could you repeat the question?
Yeah.
He wants to know what are the systematic changes you're referring to.
So any change that can be blocked pharmacologically or predicted by some kind of physiological or behavioral change is a systematic change.
Or is affected by the environment?
Yes.
Okay.
Yeah.
Okay.
So something that can either be statistically explained or manipulated actively. Okay. So in order to understand this, my then postdoc, Dhruva Raman, built a very simple model. Okay, so he built a model of how synaptic weights might change over time, and he decomposed the total rate of change into some systematic component. This is the sort of learning-related component, typically, and some random term corresponding to these fluctuations. And the way that he implemented systematic plasticity was he modeled it as an approximation of gradient descent in a standard neural network learning task. So in this sense, we're able to separate out learning, the contribution during learning, versus the contribution where there's no change in error in this task, but the fluctuations are being counteracted by continual learning in order to maintain the error at some steady state value. So we're going to be interested in this kind of end game. This is what we're thinking of when we look at these other results where people have monitored ongoing spine dynamics with no obvious learning going on. And here's, in essence, the result, that by modifying the ratio, essentially changing the reweighting or changing gains between the contribution of the systematic plasticity versus the fluctuations, what Dhruva found is that the steady state error is minimized approximately when the plasticity to fluctuation ratio is equal. In fact, it's actually minimized when the systematic part is slightly smaller than the random part. So across magnitude of fluctuations, this relationship holds. And interestingly, if we increase the precision, the learning rule quality, and loosely speaking, quite precisely speaking, actually, all this does is it adjusts the correlation of the systematic change with the computed gradient of the task error. So the closer it approximates the gradient, the higher the quality of the learning rule. And the only interesting trend that you see here is that the optimum shifts further and further towards favoring a smaller systematic component relative to the fluctuations. So the obvious question is, why is this the case? And I'll promise you one thing, if you don't know already, you'll kick yourself, because there is a fairly trivial way of understanding this, which gets a little bit more interesting in the case where you have pure gradient descent. Let's look at some intuition here. So what we're thinking about is in this task that there's some form of sort of noisy learning going on, and in the end game, there's just noisy maintenance in the vicinity of low error. Yet the network continues to reconfigure due to the forcing of these fluctuations. Now, in a special case where we just have one synaptic weight and we have a bowl-shaped error surface like this, if we're to start at the bottom and then decompose our weight changes into this fluctuating term and this systematic term and ask, well, what happens if we undergo a fluctuation? Well, obviously, the error will increase if we're at the bottom to start with. And then if we ask how big the systematic component should be, well, it should obviously counteract the error, but also generically, if we are close to a minimum, that tends to be when we have non-zero and positive curvature, where the second derivative is non-zero. And that means if this systematic component exceeds the magnitude of this change, it will tend to actually make matters worse, potentially. Okay? So it will not optimally compensate for this fluctuation.
In this case, excuse me, do you consider the curvature of the curve? The curvature.
Excuse me?
Are you considering the curvature of the steepness of the track?
Yes, yes. The curvature ultimately is what explains this. Yeah.
Oh,
Yes. So this is a second order effect. And for that reason, this effect only comes into effect when you've approached a steady state in the vicinity of a minimum or a local minimum, because then by definition, you are at a critical point. And if you're at a critical point where error is minimized and you're not in a completely flat error surface, there has to be some upward curvature.
but all time is moving, right? All time is never is in one point is static.
Correct, correct. But on average, if you think about the expected location of this, it's going to be in the vicinity. It's going to be in a neighborhood of this.
Oh,
So in many dimensions, of course, there are many low error states. And a really crude calculation tells you what the optimal proportion of plasticity to any fluctuation should be. If we imagine we're in a vicinity of this error surface that's not doesn't have extremely high curvature so that we can approximate it sort of locally as a plane. If we imagine being kicked off this surface, then the following the pure gradient will essentially take a beeline straight back to the surface. It will always be tangent, normal to the surface. And if you do the math and just integrate all the fluctuations around a quadrant of a circle, you find that on average, this shortcut is this ratio relative to the original perturbation, which is about 61%, which is amusing to us because a lot of these studies done in vastly different conditions seem to find that the non-systematic component, so this fluctuating component, is indeed close to around 61%. I'm not pushing that too strongly here. This is just an amusing observation.
Where does the 5 come from in the calculation? Did you get that?
You need to compute the average distance you get knocked off a curve in a random direction, assuming that you're evenly distributed across a circle.
I see.
Yeah. Okay. So the take-home message of this.
In higher dimensions? Because that's a one-dimensional.
No, that's in any dimension.
Okay.
Because. Sorry?
It just looked like was it a one-dimensional result?
No. This applies regardless of the dimensionality. Okay, so the message of this work, at least, is that at steady state, where there's no obvious learning going on in this sort of special case, the systematic component of noise-induced drift can quantify the precision of a learning rule. And it's also consistent with there being quite a large amount of residual random change going on, which could drive drift. Now, we don't think that this type of noisy turnover, first of all, it may not be noise, it may be systematic, but something we've not identified. But we think that drift actually is likely even more systematic than that. And I'm just going to cite a fun fact here, and that is that noisy perturbations are not required to generate drift at all. Online learning from a fixed distribution is sufficient. Okay. Now, what do I mean by that? Well, online learning necessarily subsamples from some putative distribution. And one can assume that sampling is stochastic, or it could systematically sample, but one way or another, it's going to generate different gradient steps each time a learning rule is applied. So if we go back to this picture and try to understand what is going on in this kind of end game, if we think of a loss level set and plot the direction of the true gradient, if you like, so this is the gradient with respect to all of the data that could be gathered on a task. And we bear in mind that in the online setting, an agent or an animal does not have access to all of the data. It only has access necessarily to a subsample. Well, that subsample is going to produce a gradient which will correlate in expectation with the true gradient, but each subsample is going to be slightly oblique to the gradient. And the effect that will have if we're maintained in the vicinity of a minimum is it will introduce tangential perturbations along these level sets. So to understand what happens when you've got stochastic gradient descent, what you need to really visualize is that motion kind of along the waterline of a lake, where the lake represents the sort of minimum error that a learning rule can actually achieve. And this is analyzed in detail in these two references, not in the neuroscience literature, incidentally. You have to dig more into the machine learning literature to see this.
Excuse me,
but we mentioned this in a recent review.
Can I ask a quick question? What sets the timescale then of representation drift in this picture?
Right, two things. So, in fact, multiple things. That's a really good question, because we're going to get to part of it in the middle part of this talk. But one of them will be the gain or the learning rate that you apply here. So obviously, the larger step I take, the higher I turn at my learning rate, the larger these perturbations will be. And also the larger the steady state error will be, in fact. And this is well known in optimization.
Mm-hmm.
The other thing that affects it is something involving the correlation between the gradient that you would compute on the full data set and the gradient you compute on a subset. Now, the smaller this subset is, the larger the fluctuations or the deviations rather could be. So there is an expression, and Chowdhury and Swato provide a really nice tight expression for this that relates this to the learning rate and the effective batch size that's used in learning. There's another interesting thing that relates to this as well, and that's the dimensionality of the task manifold. And that's the interesting one that I'm going to pick up on in a few minutes' time.
Okay, great, thank you.
Okay, so hopefully what I've convinced you is that if subject to a few assumptions, if we assume that drift is not just biology being biology and doing something weird, but that circuit configurations matter, and that if they change, that ultimately can have an effect on behavior, then drift, in as much as we can observe it during stationary points in an animal's learning, can tell us things about systematic plasticity and continual learning. So in the next part, I'm going to tell you what I already hinted at, and that's how drift can help quantify degrees of freedom in the task representation. So for this, we're going to turn back to this study of PPC. And just to remind you, this population shows marked drift over several weeks. And in this work, my then postdoc, Michael and Adriana Lobach, obtained the data set that Laura Driscoll generated in this work and analyzed it. And the way they went about analyzing it was fairly straightforward for related reasons. In fact, what we ended up doing was designing a brain-machine interface decoder for this, but that's another story. But for various reasons, we asked, how well does a linear readout perform on this population? Intuitively, if you have this type of activation pattern, then you can think of each of these neurons, so each row here is a neuron, as kind of being a basis function, right? So it's like, this is almost like a kernel representation of all of the locations along the maze. So intuitively then, if we just take a weighted readout of the activity of these cells, it should be able to give us a pretty good estimate of where the animal is. And that indeed is the case. We can not only predict the animal's position, but its speed and its view angle as well. And this gives us a nice way, a nice kind of yardstick for measuring drift, because what we can do is take a linear decoder trained on a given day and then fix that decoder and ask what its prediction error, how its prediction error varies as we take data from subsequent or previous days. And we see this very obvious drop-off in, increase in mean squared error either side of the day on which the decoder is trained.
Excuse me, in the before graph,
So the last slide.
in the last slide, yes, there any reason that the curve is like to tangential?
I'm sorry, could you repeat the
There any
question?
reason that you know that you observe the
Well, why the yellow curve is
Yeah,
like,
yeah,
a,
yeah.
like a 10?
yeah
Oh, why does it look like the tanh function?
yeah.
I mean, pi has shown up here already, and now tanh has shown up here already. So I think there's some deep numerology going on. It's all maths all the way down. You know, I think, yeah, I think the universe is trying to tell us something. I don't know. I mean, if you want a boring answer, the salient points in this task, where the animal's really attending to what's going on, is at the start and at the reward point. So there's going to be concentration of responses there, and then, something in the middle. Sorry, that's probably not a satisfactory answer.
Can I ask a question here?
Sure.
What do you mean by view angle? Are you predicting where the animal is looking?
Yes. Yeah. Sorry, I should have included a diagram of the virtual task. So the animal is head fixed, so obviously can't rotate its head, but it's mounted on a spherical treadmill. So it can move basically in a plane. It can move forwards and it can rotate its field of view. And view angle is just the angle that the animal is heading in the VR environment. So it's the angle that it's currently steering towards.
Okay, thanks.
Thank you. Thank you for clarifying that. Okay. So having this linear decoder is helpful because it actually lets us analyze drift. Trivially, we can see that drift affects decoding error. But an obvious question is, can we find a drift invariant decoder in this way? And that's kind of equivalent to asking whether all of the drift occurs in some kind of linear subspace. So the way that we do this is fairly straightforward. We just take data from multiple days, being careful to separate out training and testing data, and make a concatenated model to decode across multiple days as opposed to the single day model. And the short answer is, you can sort of find a multi-day decoder, but the more days you try to include, the worse the decoder does. So there isn't exactly a drift invariant decoder. Moreover, when we try to actually model, construct a kind of null model to account for this deviation in coding performance over time, we found the following. So I'm going to sort of slow down here and explain what's going on a little bit. So what I'm plotting here is how quickly the performance of a linear decoder drops off as a function of elapsed days. And this is the decoder that's trained across a stretch of several days. So in the data, it drops off relatively slowly. And then in two models that we constructed, where we basically just made a linear regression of a linear map and then added Gaussian noise to that linear map, so that any regression model is going to have some amount of error that we can control. And we modeled drift by corrupting that linear map with additive noise. And we did this in two ways. We either constructed a model with Gaussian corruptions of this linear map that were matched to the within-day variance, okay? And what we found was that the decoding performance dropped off far more quickly than in the data. Or we constructed a model where the sparsity was matched in a sense to the data, because the data is very non-Gaussian and you have these localized activity patterns. So in a sparsity matched null model, interestingly, the drop-off was even more dramatic. So this indicates several things. So one of them is, straightforwardly, that the drift doesn't just occur in some linear null space of a readout. It also implies that drift is far more systematic and constrained than a pure random walk would be. So these two null models, if you like, are models of Gaussian random walks occurring in a space of equivalent coding dimension. And with this model, we could adjust the dimension of this linear map. And by changing the dimension in which this random walk occurs, we can ask whether we can actually match the drift rate. So this goes back to an earlier question. And what we found was that in order to get this null model, the random drift model, to line up closely with the data, we had to reduce the rank of the drift subspace quite significantly. So where the number of dimensions matched the number of units, this null model drifted far more than the data did. And when it was reduced to roughly 20% of the available coding dimensions, that's when we found comparable drift. So this is a kind of roundabout way of saying that drift is low dimensional, or is equivalent to a low dimensional random walk. Okay. So the summary at this point, drift appears to be related to some kind of systematic plasticity and maybe continual learning. And drift appears to be constrained to a lower dimensional manifold than population activity is. And there's an obvious prediction here, and again, this was anticipated in the previous discussion, and that is that task complexity should modify drift rate. So this is something that we've suggested to our experimental colleagues that they should look into. And what I mean by that is, if we have this mental picture here of what causes drift, well, the mean squared deviation in location, in initial location of a random walk scales linearly with time, and it also scales in proportion to the number of ambient dimensions in which this random walk occurs. So under this kind of Brownian motion model of drift, one would anticipate drift rates to be far higher when the dimension of this allowable subspace is higher. Now, for a complex task, where the configuration of the circuit is more constrained, the ambient dimension of this functional subspace is smaller. Therefore, we would expect lower drift rates. So for a complex task or environment, this is what should happen. We should measure some amount of drift, for example, in the population correlation of a population vector with an initial population vector. And in that same animal, if we can somehow simplify the task or the environment, then that same circuit should show a higher rate of drift if all of this, if essentially this model of how drift occurs is true. If it is the case that continual learning is the thing that is constraining drift to occur within this subspace. So in summary, we should see something that looks like this. So this is an experimentally testable prediction of this conceptual model. And it essentially would allow researchers to figure out how much task complexity stretches the capacity of a neural population to represent that task. Okay, so we're making some progress. We've talked about how to relate drift to plasticity at a sort of low level, and then more abstractly related degrees of freedom in a task representation to the dynamics of drift. Final thing I'm going to get to is a question that we've been asking recently, and that is whether the certain features of drift, empirical features of it, if you like, are indicative of underlying plasticity mechanisms, in particular, mechanisms that are in place to cope with drift. Because we haven't really discussed that yet. If one circuit is undergoing drift and another circuit is supposed to play nice with that circuit, it's not clear necessarily how those two circuits become updated in a coherent way. And that essentially is the setup for the work I'm going to describe. So my then postdoc, Michael Rule, who's now faculty at University of Bristol, set up the question in the following way. So he supposed that, okay, there is some latent behavioral state. We could call it, you know, head direction. So it could be location on a circle or something like that. And let's suppose there's a population somewhere in the brain that has this nice sparse bump-like representation of this latent variable, but it drifts. And it's the job then of some readout brain area or set of neurons to actually extract the value of this latent variable. So suppose there's some downstream circuit that needs to integrate the value of head direction, for example, but all it has access to is this circuit that has this drifting code. How might it go about it in the absence of access to the ground truth? So quite obviously, if this circuit starts initially tuned so that there's a response of one of these readout neurons that's tuned to a specific value of this latent state, then over time, if the readout weights are fixed and this drift occurs, then essentially what will happen is that this peak of activity will die away and this cell will become unresponsive. If, on the other hand, we implement a fairly simple, biologically reasonable model of homeostasis, where the output variance of a neuron homeostatically adapts over time. So this is something that I actually saw firsthand in my PhD work. If you deprive neurons pretty much anywhere in the nervous system of input, they find a way to make themselves busy. And they do this in a number of ways. They can increase the magnitude of synaptic inputs, excitatory synaptic inputs, or they can alter their intrinsic excitability. Essentially, there's a kind of range of activity where neurons seem to converge towards, and it's non-zero. So we implemented a straightforward rule that maintains the ups and downs in activity of a neuron at some set point. And this is what happens. So essentially, for short periods of time, the peak becomes normalized. So if there's any sort of location in this latent state where this neuron is more excited, then this variance, the variance of the neuron essentially gets normalized, and then that preserves the magnitude of this peak. However, once the reconfiguration of this drifting circuit becomes large enough, then this peak will abruptly jump to another location. So hold that in your mind for the time being, because this is an interesting observation, we think. In any case, Michael wasn't happy with this, and he said, well, you know, could you do better in the usual computational neuroscience sense, where you tie your arms behind your back and you only play by the rules that biology is allowed to access, and you take another biologically plausible rule, in this case, Hebbian plasticity, and you essentially reinforce the input weights. So you treat the output of this neuron as a prediction that strengthens correlations between input and output. And what this will tend to do is maintain the readout of these units despite some amount of drift. Now, this won't work forever, okay? That would be impossible because there is going to be some some non-zero probability at all points in time of disrupting this code, but it outlasts a naive normalization and certainly outlasts a fixed readout. So in principle, even if a circuit is deprived of signals that allow it to calibrate the readout of this circuit to some ground truth of the latent state, it could maintain some kind of coherent readout despite substantial drift. So going back to the effect this simple homeostatic rule had, what we see is that this homeostasis produces what Michael called punctuated stability in the population column. So a tuning curve will remain consistent for some period of time, then jump, then remain consistent, and then jump. Okay, so it makes this drift a little bit more switchy, if you like. And this reminded us, at least anecdotally, of the data that we'd stared at. We wondered whether this can explain qualitative properties of drift that are seen in data. And then we wondered further whether this model of drift, if you like, is more reasonable and whether it might actually have nice properties that facilitate downstream decoding. And if that all seems a mystery, I'm going to explain it very quickly and hopefully clearly now. And this became the work of my current PhD student, Charlie Miku. And he asked this simple question: are there properties of drift that facilitate stable decoding despite drift occurring over time? So let's imagine we've got this population of neurons all tuned nicely to some latent variable. And we'll consider two alternative models of drift. One in which any neuron at any given moment can just nudge slightly, for example, from a Gaussian distribution. Or another one where most neurons undergo imperceptible changes in their tuning, but a small subset undergo large changes. So this is more the kind of punctuated stability model that we mentioned earlier. Now, in the long run, both models will produce a population where this representation gets completely scrambled. But the transition to this end game is very different. And here I'm just showing you the distribution of increments. So in one case, we have something that we're calling gradual drift. We have a kind of Gaussian distribution of perturbations in tuning. And another where we have sudden drift. So either the tuning change is a delta function weighted with something less than 1 at 0. So there's either no change or there's an evenly distributed change, in this case, around the unit circle, because we took the unit circle to be the space of this latent variable. And in order to address this question of whether these two different models of drift have any effect on a readout, Charlie set up a decoding problem. So he used a Bayesian decoder. So he was basically assuming the neural circuit can do the absolute best it could, where on the first day it's shown stimuli and it's shown the output of this drifting population, and it has to make its best guess of what the underlying stimulus is. But then on subsequent days, it's deprived of the stimulus and only has access to this readout population, which is now undergoing drift. And this, by the way, makes predictions about what one should see in the distributions of changes in correlations between pairs of neurons. And this is the thing that allows us to compare this to data. So after one day of drift, under the incremental drift model, we should see the distribution of correlations kind of spread out a little bit. By contrast, the abrupt drift model will always show this immediate appearance of the second peak, where essentially there's been a large correlation change away from 0. And that will become more obvious at intermediate time points. And then in the long run, these two distributions will converge. And the punchline of all this is that sudden drift in the sparse code, the sparse population code, is easier for a decoder to correct for. So what Charlie did was take fixed decoders, and unsurprisingly, fixed decoders over time will degrade to chance. And then an adaptive decoder, which has either sudden drift or gradual drift. And what he finds is that the adaptive decoder, in the case of sudden drift, always outperforms the gradual drift decoder. And this is when the rate of drift, the overall change day to day, is calibrated to be the same. Essentially, what the decoder does is it explains away changes in its output. If all of the inputs change slightly, it's harder to tell whether a few of the inputs were erroneous. If, on the other hand, a few of the inputs change dramatically, but all the other inputs are consistent, Bayes' rule will allow you to re-weight the evidence in such a way that you ignore the input from the neurons that are changing their mind dramatically and weight the more consistent inputs more highly. So an obvious question then is, does this type of drift, which type of drift actually occurs in data? Right? Because if we're imagining this putative Bayesian decoder existing somewhere in the brain or something, some kind of hack like this, then this is only an interesting hypothesis if there's some evidence for this type of drift as opposed to this type of drift here. And what Charlie did was went out to two data sets. So he took the Driscoll et al. Data set that I've already described, as well as a more recently published data set from Michael Gord's lab, which studied representational drift in the visual cortex. Now, the drift here is far slower than the drift in posterior parietal cortex. But in both cases, he detected this appearance of this initial peak that's separated from zero change in correlation. So we see this early appearance of the second component. And when he ran the numbers, essentially a statistical model that has gradual drift does a very poor job of describing the changes in these pairwise correlations. It's very, just doesn't fit the data very well. You really need this second component and a mixture of the two models to do a good job of fitting the data. So long story short, there is a component of gradual drift, and that's perhaps not surprising because there's some amount of kind of small random noise increments in many processes in the brain, but there's also evidence for this detectable sudden drift component, which might tell us two things. Number one, that this punctuated stability model is indeed something that's going on in these circuits. And number two, there might be advantages to that for circuits that need to decode from drifting circuits in the absence of ground truth from the external world. So that's all I had to tell you about. This third point, I would say, is far more speculative, but it's interesting to ask what features of this apparent drift make sense from a normative perspective, as well as trying to relate it to more fundamental physiological processes or the role of various circuits in representing learned tasks. So with that, thank you all for listening. I'd like to thank Mike for the invitation. And I must thank all of the people who did this work. So Charlie, Adriana, Dhruva, Michael were the main contributors to all of this. And we relied heavily on our collaborators in Chris Harvey's lab as well, providing data. So thank you all for listening. And I've probably got a little bit more time and energy to take a few questions if you have any.
Jean-Marc, you want to go?
I have a question. C'est Jean-Marc Felous.
Sure.
Can I go ahead? Oh, thank you. Thank you for this talk. This was amazing. My question is just I'm wondering what you're thinking about sleep. So some of the data that you've shown are across days, of course, and
Yeah.
rats and mice sleep. I mean, across multiple days they sleep, and there is memory consolidation. You know, the very
Yeah.
classical phenomenon of reactivation, and
Yeah.
this, you know, general finding that when you compare the activity of a task to that of the immediate sleep afterwards, you see this slowly decreasing match between the activity of the cells during replay and the activity during the task, which people have interpreted as being a recoding of activity due to memory consolidation, the fact that the memory integrates itself with the other memories that are already present.
Yes.
And so on. And so, you know, I never heard people talk about that phenomenon as drift. And and it could be part of it could be drift, part of it could be actual completely reconfiguration of the circuitry. And so I'm curious to hear what you thought about, you know, this phenomenon of replay, memory consolidation, drift,
So
how does it all fit together? Thank you.
I think they do fit together. So Michael, when he was studying these data sets, really wanted to ask this question, but we couldn't because, as with most data sets, they're gathered when the animal is mounted on a microscope and has its attention span available. And the other 23 hours of the day, it's playing with its buddies or sleeping. And I just don't believe that what goes on outside of the task is not relevant to how the task gets represented. For sure, the brain is iterating over previous experience. There's strong evidence that offline replay affects the ability of an animal to perform and recall. So for sure, this is going on. And the way I think of that is that we tend to think of this learning as something being an external signal. Well, that's why I include the word feedback here, because as far as the neural circuit is concerned, it's receiving inputs from somewhere, possibly modulatory signals as well. And if it's this deep circuit that's not directly connected to the periphery, in a strong sense, it doesn't know whether it's asleep or awake. And unless miraculously all the plasticity sort of switches off during sleep, which is a possibility, but I doubt it, this process is going on the whole time. And I would expect, and I'd love to see this. It's phenomenally difficult to imagine how you would study it, because by definition, to study drift, you have to look at activity patterns with respect to some measurable variable. You can look at internal configurations of cells with respect to each other. In some work that we've done, we've found that doesn't actually seem to change that much. So-called noise correlations don't really explain drift. There's a sort of genuine reconfiguration of the circuit. But yeah, I believe that sleep will have a role in this. And there have been a few recent studies that have argued actually two different ways. So there's two studies I'm aware of. One from Yaniv Ziv's group, another from Dori Derdikman's group, where they tried to tease apart the effect of the passage of time versus experience on drift. And there is definitely a role for experience as an engagement with that task in driving drift. But depending which group you listen to, time also is a factor. And then outside it, so both of those studies were in the hippocampus. And then in olfactory cortex, there's a very nice paper from Richard Axel's lab by Andrew Fink and Carl Schoonover, who monitored responses in piriform cortex. And they definitely found a role for both time and experience in drift. So there is this kind of churning over that goes on, and sleep could be a component of that. And this contributes in addition to actual contact with the task.
Thank you. Great.
In your model of homeostatic plasticity, what features of homeostatic plasticity are not included in your model?
Yeah. It's a very abstract model. So essentially all it's doing is taking like a running average of the variance of the output of a rate-based neuron, right? And it's feeding back, it's controlling the gain of that neuron, so the steepness of its nonlinearity. So it's lumping a whole bunch of things together. You could interpret this as a change in the threshold, a change in the gain, the intrinsic gain of the cell, or in fact, a change in the synaptic gain. They all kind of end up being equivalent in a model that's that abstract. So we can't really say.
Are there any features of homeostatic plasticity that can't be explained in your model, or what features are left out for salient parts of?
What features are left out? I suppose there's no. So there's one kind of interaction in homeostatic synaptic plasticity where there's this kind of heterosynaptic effect where input is rebalanced across synapses. I think under certain circumstances, maintaining variance might be equivalent to that, but that would depend on the input to the neuron. So it might not capture all aspects in all cases. But the honest answer, as you can tell, because I'm rambling, is I don't know. But it does lump together a bunch of features observed in certain neurons. Because this is the other problem, is there isn't just one kind of homeostatic plasticity. There are many of them. And they vary depending on what type of neuron you're looking at.
May I ask a maybe basic question or maybe not? Okay, so you showed these graphs where you mapped the neurons by position on the track. Now, if I'm trying to make guesses, if I'm trying to make some guesses about the actual mechanisms of plasticity and substitution of neurons, how does this drift map to the physical location of these cells which are involved in the brain or wherever they are?
Yeah, we looked at that, and the disappointing answer is there's salt and pepper scattered all over the place. So we did look in the PPC data because we had access to the raw microscope images, and we didn't see any evidence that certain spatially localized cells move together, if you like. But that's not always a good indication of whether the cells are coupled, I guess.
Okay, so now if I'm combining facts that nobody else connects, and if I'm thinking of that guy who got a lance rammed into his head and lived on, so that means that if these neurons are distributed salt and pepper and can be substituted salt and pepper, then if you punch out a large hole into the contiguous sets of neurons, that makes it robust against that. So salt and pepper is something that gives me an answer about robustness.
Yeah, salt and pepper is robust, but I wouldn't generalize from rodents to humans. Responses are far more localized in primates, at least, and I expect that that's the case in humans too. Whether that then ends up translating all the way down to detailed correlations in activity in sort of neighboring neurons, I don't know. But in mice, in cortex, lots of stuff is kind of salt and peppery with a very sort of vague spatial drop-off in correlation. And actually, Chris Harvey's lab has done some really nice work looking at that, looking at how much activation of nearby neurons influence the activity of a particular neuron in question. There is a non-zero spatial component, but it's relatively small.
Okay, that helps. Thanks.
Hi, I had one quick question about the rank finding that you have, where about 20% of the rank was needed for that random walk.
Yeah. Yeah.
In the recordings that you used, did those have both excitatory and inhibitory neurons, or was it only?
No, they're just excitatory.
Okay, I wonder if that, because that 20% finding, since it was a bit robust across number of cells, if
Yeah.
that's related to or indicating that inhibitory neurons might be kind of driving drift.
Maybe. Yeah. No, that's an interesting question. I think I see that question in the chat, actually. I just pulled it up. This has not occurred to us. I don't know. It's quite possible that interneurons, well, we know that interneurons, in a sense, have many roles, and one of them is that they can kind of scaffold the co-activation patterns of a population. And that, you know, is especially prominent in places like entorhinal cortex and part of the brain like that. So yeah, you know, we don't know what's causing the drift. It could indeed be interneurons. I guess a really good way of asking that question is to try to model drift as either changes. You know, you could make sort of two straw man models, one where it's only excitatory connections that generate drift, and one where it's only inhibitory. And if there are marked differences that you'd observe in a population, that might tell us something. It's a nice idea.
Thank you.
Can I ask perhaps a very naive question, so don't hesitate to say if it's completely stupid. But I wanted to know, what was the size distribution of the synapses involved in drift?
Oh, yeah, I don't know. Yeah,
I mean, I'm asking because there's a
Yeah.
really broad size distribution of synapses, and
there are, yeah. So it's a while since I've read those papers, and you know that, as you're probably aware, there's a generally accepted sort of rule that, you know, these large mushroom-shaped spines hang around, filopodia come and go, and then there are other spines of intermediate size that make up their mind.
right.
Yeah, I think the
Well,
only
that's why
way...
I'm asking the question exactly.
Yeah. So I think consulting the original studies is really the only way to get at that. Off the top of my head, I don't remember. But I do remember that there were other studies by... So this is going a way back. So Carlos Portela Callao, who's in, I think, UCLA, was doing optical monitoring of cells in barrel cortex, and they did find a sort of differential rate of change across spine types, is my recollection. My recollection is also that I wasn't hugely surprised by it, so it probably suggests that the big spines were more stable. That certainly is what the rest of the literature suggests.
Yeah.
So yeah, yeah, for sure. So there could be, you know, a kind of strong scaffold that maintains responses fairly consistently. And that's something that actually a few theorists have put forward as an idea. So Claudia Klopath has followed up that idea. I think for us, you know, I presented the spine turnover data simply because it is an example of a low-level change that seems quite significant. But there's no getting around the fact the neural activation, one way or another, changes significantly. Now, if that's not because the input changes, then it has to be something else.
Right.
So I'm not saying that the input is everything, but it's one of the first places I would look, I think.
Great, thanks.
Thank you.
And thanks for a very clear talk.
Thanks.
Any further questions? Thanks again, Tim.
Great. Thanks again.
Good to see you. And
You too. And yeah, I really appreciate the invite. Hope it all made sense. Take care.
yeah, have a nice rest of your evening.